unable to bind to the human ACE2 receptors.
Right, that's exactly right.
So as of 2010, scientists including Shi Zhengli in Wuhan, China, knew that naturally occurring horseshoe-back coronavirus spike proteins could not infect humans.
That's right.
Based on your testimony, it sounds like you discovered at least 2 man-made inserts into the SARS-CoV-2 spike protein genetic code that prove it was produced in a lab.
Right. The research papers that Shi Zhengli and Barrick and others published, they were very clear and somewhat proud of the fact that they'd been able to modify the virus and to insert changes into it that made it more effective and potentially more dangerous.
Is there some laboratory mechanism where they're able to take genetic code from elsewhere and put it into a virus?
Right, so you can actually build anything if you know what the structures are. It's like building a car. If you know what the parts are, you simply put it together. Many of the research studies that we have pointed out, they took bits and pieces of one virus, combined it with bits and pieces of another virus. If you go and look at those papers, you'll also find that they were very specific by inserting 5 nucleotide base pairs in there. So they simply switched the base pairs, and that will result in a different amino acid being added and a different protein.
Let's talk about the first insert that you found in the SARS-CoV-2 virus that led you to conclude that it was a man-made bioweapon. The first one I think you referred to is the furin cleavage PRRA insert, correct?
That's correct.
Can you explain briefly what that is?
Okay, so that's 4 amino acids that were put in. When this virus attaches, and I know this is long and complicated because a lot of people have heard of ACE2, which is one of the receptors, but it's a multi-step process. The HIV glycoprotein 120 insert, which was the second one we talked about, is required to attach to what's called a sialic acid receptor raft that stabilizes it and puts it in position. The virus then comes into contact with the ACE2 receptor. That then undergoes a change with something called a TMPRSS2 receptor, which is critical to understanding because that begins the process of bringing that virus into the cell. It then has to undergo furin cleavage at the furin cleavage site, and that's the PRRA, which makes it particularly infective for human cells. And then it comes across also something known as the neuropilin-1 receptor, which is found in the brain.
How did you discover that this PRRA furin cleavage insert had been made to SARS-CoV-2?
All you have to do is the nucleotide-based sequencing, which is how we know all the variants occur. And when you do that, you find this PRRA insert.
Now, this genetic code for the furin cleavage insert, the PRRA, is that found in any other coronavirus in nature?
None.
It's not found in any other coronavirus that exists in the wild?
Right. There's absolutely no other coronavirus that has this PRRA insert. And that's 12 nucleotide bases, not a single base like sickle cell, but 12 specific nucleotide bases to provide for 4 amino acids that very specifically is connected with the furin cleavage site, which the U.S. government owns the patent on.
So the fact that this insert and this genetic sequence for this insert is not found in any other coronavirus in nature, what did that tell you?
Well, since mutations occur 1 nucleotide base at a time, you would have to come up with some phenomenal explanation for why 12 nucleotide bases suddenly inserted themselves into a virus that none of the other viruses in that category have. The original premise that was put forward and actually published in The Lancet was that this was a naturally occurring virus with an animal host that got transmitted to people, but yet there's not been a single animal model found with this. Neither has there been a single coronavirus that has this PRRA insert.
You mentioned the second insert, an HIV glycoprotein.
Right.
So are you saying that sections of the genetic code for HIV were inserted into the SARS-CoV-2 virus?
Yeah, Shi Zhengli published this early on many years ago, where she was very proud of the fact that she did this. She used an HIV pseudovirus to do it. And we also know that HIV glycoprotein 120 is a prion.
So this is the HIV that became famous in the 80s and 90s. It's that HIV.
Right.
And somebody took pieces of the genetic code of HIV and put it into SARS-CoV-2?
That's what Shi Zhengli admitted to in publications that she had, yes.
Were you able to discover why that was done?
According to the papers that she published, it was to increase the infectivity to see if she could make it more infective. But it's even clearer that what she was going for was the development of a prion disease, because where everybody is kind of coming up to speed with a virus and how challenging it can be to deal with, what's even more challenging to deal with are prions, because prion diseases are relatively new in our understanding for treatment, and they cause permanent damage unless they can be reversed. And so the development of a prion, putting HIV glycoprotein 120 in there, attaching to the sialic acid receptor, there's nothing about that that is advantageous for people. And the ability to produce prion diseases is quite a weapon.
And how lethal are prion diseases?
People die from prion diseases.
Is this process of inserting these amino acids to increase the infectivity, is that gain of function?
Right, so gain of function is anything that you do that changes a naturally occurring biological agent like this virus. And so the premise of gain of function really has a good premise behind it. The original idea being that if you could stay a step ahead of a potential infectious agent, say a coronavirus, if you could stay just a step ahead, you could do a good job of providing treatments for people and knowing when and where it was going to spread. This isn't a step ahead. This isn't a single nucleotide base. This is a spike protein that has an HIV glycoprotein 120 inserted into it. This is a spike protein that has 4 amino acids to make a furin cleavage site, the PRA insert. This is a spike protein that has multiple other, according to Luc Montagnier, HIV and SIV inserts. This is not something that evolutionarily evolved. The evolution of something with this many changes would take hundreds or thousands of years. This is not staying a step ahead of it. And making this provided no advantage for human beings, which makes it a Biological Weapons Convention treaty violation. It's playing God.